Buenos días a todas las personas que nos acompañan. Bienvenidas y bienvenidos al segundo día de Jornadas Académicas en el marco del 60 aniversario de la fundación del Departamento de Fisiología, Biofísica y Neurociencias. Today, we are honored to have a world-renowned scientist, Professor Henriette van Praag. I'm going to ask Dr. Carmen Vivar to introduce our distinguished guest. Professor van Praag, thank you very much for having accepted the invitation on the occasion of the celebration of the 60th anniversary of the Department of Physiology, Biophysics, and Neurosciences. Dr. Carmen Vivar, please.
Thank you. Good afternoon, everyone, and welcome to today's keynote lecture on the occasion of the celebration of the 60th anniversary of our department. Today, we have the opportunity to learn from one of the most accomplished contemporary scientists, Dr. Henriette van Praag, who have made an important contribution about the influence of exercise on the brain and behavior. She recently identified some peripheral factors from tissue outside of the brain that may play a role in the beneficial effects of exercise and learning and memory. Dr. van Praag is an associate professor at the Charles E. Smith College of Medicine and Florida Atlantic University Brain Institute. Her research aims to understand the underlying mechanism of the beneficial effect of exercise on brain function and behavior. Dr. van Praag received her PhD from Tel Aviv University in Israel, followed by a postdoctoral training in neuroscience and cell biology at Robert Wood Johnson Medical School, where her research on nerve growth factors led to an interest in the brain plasticity. She then served as a staff scientist at the Salk Institute for Biological Studies in La Jolla, California, when she discovered that voluntary running in rodents increases the number of new neurons in the hippocampus, a brain area important for learning and memory. Her successful research landed her to start her own research group at the National Institute of Aging in 2007, where I had the great pleasure or the great opportunity to join her research group as a postdoc. In 2018, she moved her laboratory to Florida Atlantic University. Dr. van Praag has received numerous awards throughout her career, including the NIH Merit Award for Mentorship of Women Scientists in 2016. Additionally, she currently serves as a co-editor-in-chief for the journal Brain Plasticity. We are delighted to have her virtually here today to share her expertise with us. Please help me to welcome Dr. Henriette van Praag. Henriette.
Thank you, Carmen, Dr. Vivar, for that very kind introduction. I really appreciated it, and I'm happy to be here for the festivities of the 60th anniversary of your department. And so thank you very much for inviting me here today. So I can start to share my screen as usual. Is that the...
Yes, please.
Okay. So yeah, you can see my screen, I hope.
Yeah, we can see it.
Okay, thank you. So yeah, this just, as Professor Vivar already mentioned, I'm located in Jupiter, Florida. This is, I just want to share a brief photo of our campus. I'm in the Stiles Nicholson Brain Institute, and we are right next to Scripps Florida Research Institute, and also the Max Planck Florida Institute. So we have a nice small campus here with a lot of good neuroscience going on. So today I will be speaking about the role of muscle in the effects of exercise on neurogenesis and memory function. However, before I go into that topic, I want to give you just a background on some of the work that we have done previously, including Professor Vivar's contributions, important contributions to our research on understanding neurogenesis within the brain. So for the first part of the talk, we will stay within the brain, and later we will expand to the periphery, and particularly with a focus on muscle. So as you all know, neurons are essential for cognitive, emotional, motor, and sensory function. One of the most important brain areas that we have in our human brain is the hippocampus, which is essential for the formation of new memories. Unfortunately, this is also one of the first brain areas, together with the entorhinal cortex, that is affected by neuronal loss in Alzheimer's disease and other neurodegenerative conditions and dementias. However, we now also know that this is one of the few brain areas that can give rise to new neurons after birth. And this is not something that was always well accepted. Indeed, the famous neuroanatomist Santiago Ramón y Cajal stated in 1928 that once development was ended, the fonts of growth and regeneration of the axons and dendrites dried up irrevocably. In adult centers, the nerve paths are something fixed and immutable. Everything may die, nothing may be regenerated. However, he also added that it would be up to future generations to reverse this harsh decree. And indeed, we now know that there are two brain areas that can give rise to new neurons. One is the lining of the subventricular zone of the lateral ventricle gives rise to new neurons in the olfactory bulb. The other is the subgranular zone of the dentate gyrus of the hippocampus, which can give rise to new hippocampal granule cell neurons. And this initial discovery was made by late Joseph Altman and his colleague, Shirley Das, in the 1960s. However, this work only became really well accepted much later in the 1990s, when new technologies became available to label dividing cells and to study their progeny. So the two main methods, or two of the main methods that I use to study adult neurogenesis are depicted here. One is BRDU, bromodioxyuridine labeling. BRDU is a thymidine analog that is taken up by any dividing cell in the body or brain. And this marker can be combined with a marker for neurons, in this case, the neuronal marker nu-N. It does only allow you, though, to see the cell nucleus. So another method that we used, where you can see the entire morphology of these cells, is using retroviral labeling, where you have a fluorescent reporter that is expressed within a retrovirus that labels the entire cell body and its processes. So so BRDU, that's the first one that I mentioned, the thymidine analog. You can inject it intraperitoneally into mice, and it will label dividing cells. And here you can view these immunocytochemically. So this is a DAB reaction and coronal section through the mouse brain. And this allows for easy quantification of these nuclei of newly generated cells. And you can also determine whether these cells have become neurons by looking for double labeling between a neuronal marker, such as this one here now in green, nu-N, and BRDU in red. And these orange tinted cells are considered newly born neurons. However, at the time when this method was used, the first papers were published using BRDU labeling. For example, Kuhn et al. In Journal of Neuroscience in 1996. There was some pushback in the field because BRDU can also be taken up by cells undergoing DNA repair. So to further validate whether these cells are indeed newly born neurons, we developed this retroviral labeling method to really study how these cells proliferate, migrate, and differentiate into new neurons in the adult brain. And this is, as you can see, a fairly slow process. It takes about a month for a newly born neuron to go from a proliferative state into a fully differentiated new neuron. So to label these cells, we make a very high titer retrovirus that expresses a fluorescent reporter, which can be injected directly into the brain. It can be visualized without any processing, so that it allows for electrophysiological analysis and also for histological processing. And then you can really see how these cells develop from this sort of early shape, triangular shape, over time into full-fledged new neurons. So this is just to give you a little bit of the background on neurogenesis and some of the methods that we use to study it in vivo. Obviously, these cells, these newly born neurons come from somewhere, so they actually arise, as I already mentioned, from residual stem cells that are present in the lining of the lateral ventricle and in the subgranular cell layer. These so-called progenitor cells can give rise to different types of neurons, but also to astrocytes and oligodendrocytes. At least that's been clearly shown when you grow these cells in culture. So this is a neuroprogenitor cell culture stained for DAPI of cells that are kept in a proliferative state by adding factors such as EGF and FGF. And then if you change the conditions, for example, by adding forskolin or retinoic acid, these cells can be induced to differentiate into either neurons. This is the neuronal marker TUJ1, oligodendrocytes, RIP, and astrocytes. This is GFAP positive cells. So basically, in order to generate these differentiated neurons, the adult neural stem cell, we now know, gives rise to an intermediate precursor cell, which is called transiently amplifying cell. So this is in vivo. And these cells retain the ability to divide, and their cycle is much faster than those of the actual stem cells. and they divide asymmetrically. So after each cell division, a transiently amplifying cell will give rise to a post-mitotic daughter cell, and then another amplifying cell that re-enters the cell cycle for an additional round of asymmetrical divisions. So this is just again to show you a brief overview in this picture of the process. So this sort of triangular cell that you see in green, those are what we call the quiescent neural progenitor cells. They will express markers such as GFAP, SOX2, nestin. They don't divide that frequently, but they give rise to the amplifying neuroprogenitor cells that then become committed neuroblasts type 1, first type 1. And when they become a neuroblast, they start to express early neuronal markers such as PSA-NCAM, double cortin, beta tubulin. And then over the course of several weeks, they mature into fully fledged neurons that express mature neuronal markers such as nu-N and calbindin. So this is, again, this is a unique process that happens in the dentate gyrus of the hippocampus. And it also happens in subventricular zone, but it's this slightly different mechanism that I won't go into today, because our focus is really on the hippocampus. Why the hippocampus? Well, it's important for learning and memory, and also because hippocampal neurogenesis in particular is unique in that it can be very strongly regulated by extrinsic factors, such as behavioral changes, such as exercise. I'll show you increases neurogenesis, whereas aging or exposure to stress, high-fat diet, excessive alcohol intake will reduce the number of these newly born neurons. And this happens against the background of genetics, growth factors, hormones, neurotransmitters. Different mouse strains have different rates of neurogenesis and proliferation. Growth factors, as I'll show you, such as FGF and BDNF, are important for the development of these newly born neurons. Hormones, stress hormones such as corticosterone have a negative regulation, whereas hormones such as estrogen and testosterone are positive regulators. And then also neurotransmitters, particularly the monoamines, play an important role in the development of newly born neurons. So the first study showing that there could be an upregulation of the number of newly born neurons, done by Gerd Kemperman, and he was a postdoc at the Salk Institute. We overlapped during that time, and he showed that if you house animals in a larger cage, an enriched environment with more opportunity for social interaction, learning, and activity, there's also more neurogenesis, more new neurons are born and survive. And these animals also show improved cognitive function. However, we asked the question, which component of the enriched environment really enhances neurogenesis? And this is actually a later study that we did in 2010 that I'll show you, where we housed animals either under control, running conditions, enrichment only, so meaning toys only, or enrichment and running conditions. And again, we injected the animals with BRDU for the first 12 days of the study to label dividing cells. So this is the setup. You can see control condition, running condition, toys only, enrichment plus running. And I just want to show you this finding, because I think it's very important to realize that really only when running is included that there is this strong doubling or tripling of the number of newly born neurons. Not just with what you would call maybe cognitive stimulation toys only. So in addition, this increase in neurogenesis is accompanied by a substantial increase in brain-derived neurotrophic factor with exercise in the hippocampus. So what we can see here is that really exercise enhances adult hippocampal neurogenesis, depending on, again, the strain, the age, the conditions. We generally will see a 2 to 3-fold increase in cell proliferation, differentiation, and also integration of these newly born neurons. So next we asked, well, does exercise also improve learning and memory? So here you can see the mice housed either with a running wheel or in a standard cage. And then again, they're injected with BRDU and they undergo a test, the Morris water maze. And then afterwards we studied what the effects were on the histology and also on synaptic plasticity. So I'll just briefly summarize this for you as well. So the water maze, it is a standard mouse cognitive test. I'm sure many of you in the audience are familiar with it. The mouse has to find a platform that's hidden under the surface of a pool that's made slightly murky with non-toxic paint. And initially they cannot find it very well. There's a 60-second cutoff time. But after about a week of training with 4 trials per day, most mouse models will do quite well and find the platform. They make an internal map based on the cues in the room. So here you can see a photo of, first of all, the running mouse and also of the room where such experiments are conducted. And again, not we, but also others have shown that if you house mice for a month with a running wheel, and it doesn't matter if they are young, middle-aged, or aged, they will learn this task more quickly, and they will have a more direct pathway to the platform. Basically, they will outperform their sedentary litter mates or counterparts. However, this water maze is a very, what we call generally hippocampus dependent task. That means that if you make a lesion anywhere in any subfield of the hippocampus, whether it be area CA1, area CA3, the dentate gyrus, there will be a deficit in performance in the Morris water maze. So with that in mind, and as the interest in hippocampal neurogenesis grew, and also the phenomenon became more mainstream and well established, researchers started to look for tests that would more specifically interrogate the function of the dentate gyrus as a subfield itself as compared to area CA3 or area CA1. So one of the proposed functions of the dentate gyrus is pattern separation. So the basis for pattern separation is that there are fewer cells in entorhinal cortex than in the dentate gyrus per se, and that therefore information becomes more refined, more spread out as it comes into the dentate gyrus, allowing for the process of pattern separation, our ability to distinguish between very similar events or stimuli. So of course, we wanted to see if running will improve the ability to distinguish between highly similar events or stimuli. So for this, we used a touchscreen system in which a mouse learns to touch a screen for reward. And as you can see, the icons on the screen are very similar, 2 white squares. So this is not an easy task for a small mouse to learn, but they can do it in these special boxes that we call the touchscreen apparatus that was developed by Tim Bussey when he was still at Cambridge University. So as I mentioned, we asked, is enhanced neurogenesis associated with improved pattern separation? So we have 2 conditions here, either a difficult one where the 2 icons are small, spaced close together, it's called the small separation, or an easy condition where the 2 icons are far apart, we call the big separation, so big versus small. And we hypothesized that the newly born neurons would only become really important when this separation is small, when the task is difficult. And indeed, that is the case. So when the separation is big, you don't really need new neurons. Maybe both the controls and runners do equally well. But when it is challenging, when the separation is small, the runners have less trials to criterion, they do better than the sedentary animals. And there is also a trend towards a correlation between the number of trials to criterion and the performance on the task. So what I've shown you so far is that running increases cell proliferation, neurogenesis, neurotrophin levels, and different kinds of learning and memory, in particular pattern separation processes. However, we still do not really understand the contribution of these newly born neurons to learning and memory. Is it just the newly born neurons that are increased, or is there something else associated with this increase in newly born neurons? For example, changes in the network of new neurons. Could that contribute? And I will just briefly touch on this. This was seminal work that was done by Professor Carmen Vivar when she was in my lab. I was fortunate that she was in my lab and embarked on this very challenging project to study the inputs to the newly born neurons using a combination of viral vectors as neuroanatomical tracers. And so basically the retrovirus is modified to express an avian TVA receptor. And then we use rabies virus as a neuroanatomical tracer. And again, this is the process. I just go over it briefly. So the retrovirus expresses GFP, TVA, rabies glycoprotein. And then a month or 2 months later, we inject the rabies virus pseudotype with an avian envelope to trace the connections to these newly born neurons. These are the starter cells. This is where the tracing starts. So the retrovirus is nuclear. You can see the GFP expression in the nucleus. And then these are the rabies labeled processes. This is just one example of her work showing that several different types of interneurons innervate newly born neurons. And this allowed us to, this method allowed us to really map out the distribution of the inputs to these newly born neurons and how this evolves during the maturation of newly born neurons in the adult brain. For instance, she showed that initially the connections are local, mostly confined to the hippocampus, and later on there's strong input from entorhinal cortex, in particular lateral entorhinal cortex, which is considered to play an important role in pattern separation processes. So it's very interesting that she discovered that that is a predominant input to the newly born neurons. And then, of course, we asked, does running change the inputs to new neurons? So again, the same process using these viral vectors. And indeed it does. There's a strong upregulation of input from the caudal medial entorhinal cortex and lateral entorhinal cortex with exercise onto the newly born neurons. And if you remove this input, if you make a lesion in lateral entorhinal cortex, this will impair pattern separation, again, on the small separation. And again, as I showed you, the increase with exercise is associated with improved performance in pattern separation tasks. So basically, running does not only increase the numbers of the newly born neurons, it also rewires their entire circuitry. So what I've shown you so far is that running increases adult hippocampal neurogenesis. It changes the connectivity and plasticity of the network of these newly born neurons. And it improves learning and memory, in particular, as you can see down here, the little mouse below, its ability to distinguish between two very similar objects or events or stimuli. However, all this work that we did still did not answer the question, or began to answer the question, but did not give us a real comprehensive picture of why exercise improves cognitive function. So obviously I showed you the effects on neurogenesis. There are also changes in neurotrophin levels, as I mentioned, FGF, BDNF, growth factors that can support the growth of newly born neurons. There is changes in blood flow in the brain, changes in synaptic plasticity, neuronal spine density, and multiple neurotransmitters are also changed, upregulated for the most part by exercise. And over the years, there's been many mechanistic studies within the brain. I'm just showing one of them. This is a very nice study out of Luis Parada's lab, showing that the role for the BDNF receptor, track B, in running-induced neurogenesis, where they made a mouse model where track B is specifically knocked down in neuroprogenitor cells. And then if you have the animal running, there is no longer an increase in adult neurogenesis. So this underscores, of course, the role of BDNF and growth factors in adult neurogenesis, but still these type of studies address mechanisms that are within the brain. However, we became very interested in looking at underlying mechanisms that involve organs outside of the brain. For example, as I'll talk about muscle, we asked, is there maybe an endocrine link between muscle and brain? So we embarked on these studies based on several pieces of research that started to come out over the past years, starting in about 2008. So I'll briefly give sort of a background of what led to our research in this realm. So some interesting studies that came out were called parabiosis research. So this is a linking of the bloodstream of two mice for, I guess, for a period of time. And what Salvaleda and his colleagues showed was that if you link the bloodstream of a young mouse to an old mouse, then that will decrease the number of newly born neurons in these young animals, suggesting that there is some factor that is in the blood that influences the brain to regulate adult neurogenesis. And more recently, the same group showed that if you collect the plasma from running mice and you inject that into a sedentary mouse every 3 days for about a month, so matching our 1 month time course of development of newly born neurons, there is an increase in adult neurogenesis. And there's also an improvement in learning and memory in fear conditioning tests, and also in the Morris water maze. So this suggested that there are indeed blood-borne factors that can regulate exercise-induced neurogenesis. So then, of course, the question arises, you know, how does this happen? Which organs might be involved? So that is one way that led, or one path that led to our studies. Another interesting research study that came out around a little bit earlier, actually, than the parabiosis study, was research identifying the transcription factors in muscle that are important for contraction, and also show, and then the use of compounds that activate the same pathway, and studying their effects on endurance. And then together with that, the idea indeed that, as I'll show you, that there are peripheral factors that are produced by skeletal muscle, by liver, by fat tissue, that are secreted during exercise that can have effects on the brain. But first of all, these compounds activated a master regulator, AMP kinase, in muscle. So AMP kinase is very important for energy balance, glucose homeostasis, mitochondrial function, and it's a master metabolic regulator. And you can activate it using an agonist called ACAR. And Ron Evans' lab at the Salk showed in 2008 in a paper in Cell that if you inject mice for a period of time with this compound, and then you place them on a treadmill, they have increased endurance. So just by activating these muscles pharmacologically. So then postdoc in my lab, Tali Kobio, asked, well, does ACAR also mimic the central effects of exercise on BDNF, neurogenesis, mood, cognition? And interestingly, it does. Actually, a brief treatment with this compound can increase the levels of hippocampal BDNF levels, not to the extent that running does, but there is a substantial upregulation. ACAR also enhances spatial memory and neurogenesis in young mice. So this is in young mice. So there's an increase in BRDU labeling. And in the water maze, in the probe trial, when this is a final test where the platform is removed and we study how long the animals search for it where the animals search during a 1 minute trial. The ACAR treated animals perform stronger. They show a preference for the platform location. In addition, in aged mice, we did a similar study because with the idea that not everybody can exercise during aging, and this compound might be important for those who are precluded from exercise due to aging-associated frailty or illness. And in this case also, there is a benefit for motor activity in the open field. There's increased endurance on the rotor rod. And again, these mice do better in the water maze. Interestingly, when we used a transgenic animal in which there was a knockdown of the muscle alpha-2 AMP kinase receptor, AMPK receptor, those animals did not do well on the Morris water maze learning task or probe trial, suggesting indeed that the benefits of exercise of this compound may be mediated by muscle. I have to mention also that it barely crosses the blood-brain barrier. So that was another reason for us to think that the muscle activation was key. So activation with ACAR enhances neurogenesis and memory, and we hypothesized then that the benefits of exercise for cognition might originate from muscle. So which factors could be released into muscle that could affect brain plasticity? So to test this, we used several different approaches, but the main idea was to take rat L6 myoblast cells, treat them with the same compound or with vehicle, and then study the conditioned media. So in this particular first approach, we used an HPLC analysis of the ACAR conditioned media and looked at differentially expressed peaks, and then took those fractions and put them on neuroprogenitor cells. And we found that there was an increase in expression of neural markers such as TUJ1 and double cortin, suggesting that there is something within the conditioned media that is affecting neurogenesis. And this is, again, one example of a factor that was in that conditioned media that was later analyzed by mass spectrometry. This is called glucose 6-phosphate isomerase. And we found that when we took this recombinant protein and put it on progenitor cell cultures, it increased both neuronal and glial differentiation. GPI is something we haven't very much followed up on it. I just want to mention it. It has an interesting extracellular function as a neurotrophic factor. It's also called neuroleukin. It's also associated with malignancies, and then it's called autocrine motility factor, where it again stimulates cell growth and motility. And also it is upregulated by in muscle by exercise. However, the more interesting factor that we are still working on at this time came from a little bit different approach. Again, this was work done by Dr. Hao Mu when he was in the lab. And again, he took the conditioned media from ACAR or vehicle-treated muscle cells, placed it on the, as I showed you, on neuroprogenitor cells, and we see an increased differentiation into neurons of these neuroprogenitor cell cultures for the marker TUJ1, as I showed you also these cultures at the beginning of the presentation. So to analyze what was in the total media, not just in a fraction, he ran this out on a gel, cut out differentially expressed bands, sent it out for mass spectrometry, and then based on, we received back a list of candidates, and based on protein size and whether the factor had been present in a microRNA data analysis as a result of exercise, and whether it could be secreted. He identified the myokine Cathepsin B, a lysosomal enzyme. Then went back in vivo and showed that this protein and also its gene expression is upregulated with exercise, specifically in the gastrocnemius muscle. So these are the runners as compared to the sedentary animals. And we see that running increases in blood, in plasma over time. So not acutely, not after 3 days of exercise, but after 2 weeks, and especially after 1 month of running, there's a substantial upregulation of Cathepsin B in blood. We also studied the behavior of these mice in Cathepsin B knockout animals. So these are whole body knockouts, and they were housed under either sedentary or running conditions, tested in several tests in open field, rotarod, elevated plus maze-4, anxiety, sucrose test, and of course, our favorite Morse water maze. And what we found was that this knockout precluded the effect of running. So the mice no longer showed a good preference for the platform location, whereas in the probe trial, whereas the wild type runners did, they showed a strong preference for platform location. In addition, the neurogenic response to exercise, so the increase in new neurons that we typically see with running in wild type animals, was no longer present in this whole body knockout. So I have to emphasize, this is a whole body knockout. There are some other issues with these animals. They also show some increase in anxiety. Patch clamp recordings from hippocampal granule cells show that there's a decrease in the frequency of miniature iPSP currents, synaptic currents. There's also some decrease in levels of brain cholesterol and a decrease in levels of P11, a multifunctional protein. I'll touch back on that in a minute. But that said, when we look at the hippocampal progenitor cell cultures and we add recombinant Cathepsin B, there's an upregulation of double cortin, there's an upregulation of BDNF, showing that this factor likely does play an important role in adult neurogenesis. So, and as I mentioned, one of the ways we think cathepsin B is regulated is by P11, a calcium binding protein that's expressed in the hippocampus, and that it's downregulated in the knockouts. And also, if you use a siRNA against P11 and add cathepsin B, recombinant cathepsin B to hippocampal progenitor cell cultures, then there is no longer an increase in double cortin. Another question we asked is, does exercise increase cathepsin B plasma levels in other species, not just in mice? So for this, we collaborated with Emra Duzel at University of DZNA in Magdeburg, Germany. And he sent us blood samples of people that had been either couch potatoes, students that were walking twice a week at 50% heart rate, or people who underwent strenuous training for 3 times a week at 70 to 90% heart rate for about an hour for 4 months. And what we found, first of all, is that increased fitness is associated with elevated cathepsin B plasma levels in humans. So the red dots you see are the training people, and their change in their VO2 was correlated with their change in cathepsin B levels. In addition, these people did better on a hippocampus-dependent memory task. This is a task where you get presented this complex figure, you copy it on a piece of paper, and then a half hour later, you have to redraw it again from memory, which is quite difficult. People who have been exercising and who had good changes in cathepsin B also did well on this task. Then, more recently, we became interested in the role of cathepsin B during aging. There have been some studies showing that cathepsin B has anti-amyloidogenic and neuroprotective effects. And also that in Alzheimer's disease patients, in their brains, the levels of cathepsin B were lower than in normal aging controls. However, there are some other researchers that suggest that cathepsin B is pro-amyloidogenic and toxic for, and actually leads to Alzheimer's disease. So this is somewhat a controversial topic, but we were fortunate to collaborate with Professor Ozioma Okonkwo at the University of Madison, Wisconsin, to be able to ask the question whether exercise increases cathepsin B levels in older adults who are at an increased risk for Alzheimer's disease, either because of they have a close family member with the disease, or they are ApoE positive. Average age is 65 years of age, and they underwent a closely supervised treadmill training intervention where they gradually increased their heart rate over time. So the intensity increased, and they did this for 6 months. In these people, there was an improvement again in cardiorespiratory fitness and also in performance on hippocampus-dependent learning and memory tasks. So again, we were, this was, the human work was done in Wisconsin, but we received blood samples from these people, either that were taken at baseline and after 6 months. And we measured the levels of cathepsin B and BDNF. Other researchers looked at Klotho, an aging and longevity factor, and there was also a metabolomics analysis done by Metabolon. So similar to our initial study, we saw that there was an increase in Cathepsin B levels in these older adults after 6 months of training. BDNF, on the other hand, decreased with physical activity in the blood, and there was no change in Klotho. So I just wanted to remind you that I had said earlier that. So should I stop?
No, go ahead.
Okay. So sorry. So this is in the periphery after prolonged training. Other people have shown that as well. So I just want to make sure that you understand there's a distinction between what happens in the brain and what happens in the periphery with BDNF measures. So these levels that we show in the increase is in the hippocampus. The reduction was in the blood. Another interesting finding that came out of the study was that Cathepsin B, changed Cathepsin B levels was associated with changes in hippocampus-dependent cognition, and not the two other factors. So this is the California verbal learning test, where people have to remember a list of words. And there was a correlation between the changes in Cathepsin B and performance on this test. Then, as I mentioned, some of these blood samples were sent to Metabolon for metabolomics analysis. More than 800 metabolites were measured in this study. Of course, I won't go through all the list of them. I just want to highlight that those plasma lipid metabolites that are implicated or strongly implicated in, for example, the conversion from minimal cognitive impairment to Alzheimer's disease, are lowered by exercise. So these people showed a reduction in, for example, in the levels of sphingolipids, and also in the levels, as I'll show you, of ceramides. And very interesting and curiously, we don't really understand this quite yet, is that there's a strong correlation between the changes in these metabolites and BDNF in plasma. Very extremely strong correlations. And yeah, so we see, and the other main finding is that we see a reduction in these plasma lipids that have been implicated in Alzheimer's disease. So what we show here is that enhanced physical activity in middle-aged adults that are at elevated risk for AD have an increase in plasma Cathepsin B levels. There's a correlation between CTSB and memory. The other two markers were either lower, BDNF was lower, Klotho was unchanged. And there's a lowering of lipid metabolites that are implicated in AD. So we think together this panel could be a biomarker set to study in a very non-invasive way the effects of exercise on the brain. So yeah, so just to summarize this a little bit more for you, and also to expand this beyond what I just showed you with regard to Cathepsin B, because it surely is not the only factor. There's factors from muscle, as I mentioned, so muscle, irisine, IL-6, Cathepsin B, from liver, IGF, adiponectin has also been implicated in neurogenesis, neurotrophin levels in the brain. So there's an increasing list of circulating factors that derive from different organs that affect neuroplasticity, neurogenesis, learning and memory. As I told you about CTSB, there's also an important role for irisine, for lactate, IL-6 from muscle, the liver, IGF, beta-hydroxybutyrate out of Moses Chow's lab, and then out of Tony Weiss-Coy's lab, 2 factors, clusterin and GPLD1. And Sonata Yao and Ko-Fei So have described the effects of adiponectin, fat-derived factor on neurogenesis. So, oops. Oh yeah, and this is just, I won't go into this too much. This is another factor, irisine. If you delete it, they find similar effects to ours. I won't go through all the details. There's also a lack of an effect of exercise on learning and memory. When you delete irisine, it impairs cognitive function. And also it results in abnormal development of hippocampal neurons. I just wanted to show you that our factor is not the only one that, first of all, that is relevant. It's probably one of multiple factors that affects learning and memory, neurogenesis, brain function. So all these work together in a sort of a concert, probably, to regulate brain functions. So I guess I will stop here. I just want to again conclude the things that I told you today. So running increases neurogenesis and memory function. Exercise expands and changes the inputs to newly born neurons. To some extent, the benefits of exercise may originate from peripheral organs. I've talked about muscle, but other organs are also important. Certainly other factors that influence the brain remain to be discovered. The one we discovered, as I mentioned, is Cathepsin B. And then last but not least, I want to acknowledge the people who did all this work. Of course, Professor Carmen Vivar, her beautiful work on identifying and analyzing the connections of the adult-born neurons and how these are changed by exercise. Hayo Moon, who is also now has his own laboratory at Seoul National University with the work on Cathepsin B. And of course, all the support of the other postbacks and postdocs that were in the lab. And nothing could have been beautifully visualized without our Linda Kitabayashi, who is always helping us with the photomicrographs. So thank you very much.
Thank you very much, Henriette. A very interesting talk, and the session is open for questions. I think Lorenza has a question. Do you want to do it directly? Is Lorenza there? If not, because she wrote in the chat, what is your opinion of taking metformin, an activator on AMPK, on daily manner by adults older than 16 years old of age?
Yeah, I metformin?
Yes.
Yes. Yeah, I know it's studied now. Well, obviously it's used in diabetic patients to lower, blood sugar levels, but and also it is being studied as a a longevity factor. However, I would feel a little bit concerned because of the high similarity between ACAR and metformin in terms of the pathways that are activated through the AMP kinase pathway, because we find that if you give ACAR for a prolonged period of time, like if we treat the mice for 2 weeks rather than 1 week when they are young, it results in upregulation of inflammation, inflammatory cytokines in the hippocampus, and there is no longer an increase in neurogenesis or BDNF. So I would be a little bit concerned, or maybe more research would be needed to more carefully also look at the effects on the brain of these kind of compounds. I'm not sure that has been thoroughly investigated.
Okay, thank you.
Any other question? In the chat, I will check. Well, Luis in YouTube say a beautiful and enlightening conference. Thank you very much. Julio Morales say excellent presentation. I think Marta has a question. Can you ask directly, Marta?
Yeah. Thank you very much. Really very interesting conference. Exercise is a good stressful situation, no? And is there any information about the effects of glucocorticoids, endorphins that are released during exercise on neurogenesis?
Yeah, so people have injected corticosterone and show that will reduce, so stress hormones will reduce neurogenesis. But you're right that with exercise, there's also an increase in cortisol and is that what you're referring to? The increase in cortisol or corticosterone levels with exercise?
Yeah.
Yeah, that
has been demonstrated, no?
Yes, absolutely. That is correct. As far as I can see, that increase or that change does not affect the neurogenic or positive effects of exercise on the brain, if that is what you are referring to.
Thank you.
Jorge has another question.
Thank you for this excellent conference, Professor van Praag. I was very, very pleased to and delighted to hear. So exercise is associated with improvement in separation tasks, which is one of the hippocampal dependent memory. Mm-hmm. But how this reflects in higher cognitive functions, not hippocampal dependent. I mean, another intellectual capabilities, not necessarily dependent on the
So yes. So this is something I think where the animal researchers are a little bit lacking, because those are functions that are more difficult for us, of course, to address and model than the spatial navigation and pattern separation. But there's quite a big literature from human exercise researchers, such as, for example, Arthur Kramer, and Charles Hillman. And they have studied executive function, which is mediated by the prefrontal cortex, and planning, judging, those kind of what you would consider higher cognitive functions, and those are significantly enhanced by physical activity. Actually, those were the functions that they studied initially in humans, mostly the prefrontal cortex related to the higher cognitive functions. And then I guess when we started with the hippocampal work, they also started to look more at hippocampus and sort of merge the animal and human literature together as, yeah. But yeah, it's a good question, but I think those functions are less well studied in the mouse models, and certainly something we should think about more, how to do this.
Thank you.
Thank you.
Gerardo, do you have a question you want to? Gerardo? No? So you're here.
I have one more question.
Okay.
It seems that stress is good for is a good strategy against aging, but I mean stress produced, like for example they say produced like by exposure to cold or by fasting. Does fasting or exposure to cold has to do something with neurogenesis?
Yeah, I read a little bit about the exposure to cold, and I think the changing, or that it affects brown fat activation or something like that. I'm not really an expert on that, but fasting, or you could say caloric restriction, has been studied in mouse models. So either the animals will get less food, or they will eat every other day, like an intermittent fasting paradigm, and that will result in an increase in neurogenesis. The issue with those studies is that when the mice are deprived of food, they become a lot more active in the home cage. So then it's difficult to sort out which variable is causing the increase in neurogenesis, whether it's the actual reduction in calories or the increase in activity. So.
Thank you very much for your excellent talk. I really enjoyed it. Thank you.
Thank you for attending.
Go ahead.
¿Quién dijiste?
Gerardo.
Gerardo, okay.
Thank you. Awesome talk.
Thank you.
Was activity of the glucose 6-phosphate isomerase necessary to increase neuronal and glial differentiation?
So we never took the experiment in vivo. So this, what I showed you, was adding of the protein just to the cultures, but we've not followed up with that. Whether is that what you're referring to? Whether that was needed in vivo in the brain?
Yes, but also if the activity of the enzyme was necessary. You know, because this enzyme has a very good inhibitor that is glucosamine 6-phosphate. Mm-hmm. And it inhibits the enzyme allosterically. And I used to work with that enzyme.
Okay.
We didn't know what was its function.
Okay. So I'm sorry, I cannot answer. I do not know the answer to that question, but I think that would be something that would be very interesting to test, and maybe use the compound or the reagent that you are you working with it in the context of neuroscience or neuro?
I was working this just in well, I was working with the E. coli enzyme at that time.
Okay.
And I was interested in the enzymatic mechanisms. But we were wondering at that time, what was the function of this enzyme?
I see.
So at this moment, the function of this enzyme is still unknown.
I see.
And it's discovered since, I think, the 50s, last century, by Leloir, Nobel Prize in Biochemistry. Probably its function is related to glycosylation of proteins, probably.
Mm-hmm.
Nobody knows for sure. So it's quite interesting.
Yes.
This enzyme related to the central metabolism has such a clear function in differentiation,
yes.
At least in vitro. It would be very nice to see if that is also the case in vivo.
Yes.
So wonderful talk.
Thank you. Thank you for your question.
Okay, Ismael. Ismael has another question, or maybe I can directly. Okay.
I like a lot your talk. So I just want to know if myelination in hippocampus could be influenced by exercise.
Myelination?
You know, myelination in the hippocampus. It could be influenced by exercise.
Yes, it's not really my, how do you say, my expertise, but others, other researchers have shown that there are effects of exercise, excuse me, both on astrocytes and on oligodendrocytes. So there are proliferative effects. There also is research in humans who are multiple sclerosis patients, which where demyelination disease, and there is some beneficial effects of exercise in these patients. I'm not sure if it's directly due to remyelination, but yeah, there definitely are effects of physical activity on glia. I'm sorry, I'm really not an expert on those effects, but they are there.
Okay, thank you.
Yeah. Dr. Balda, do you want to make the question directly?
Yes, that's okay. Many thanks for the excellent presentation. I would like to know how much exercise was needed in that studies that you mentioned, that there was the reduction in the transition to mild cognitive impairment to Alzheimer.
So those people exercised 3 times a week for 6 months. I think it was at about 70%. I have to go back and look at the heart rate. So quite intensive for about an hour, 3 times a week.
Okay. Yes, but it's not so much.
No, I think it's doable.
That is the good part. That is doable. Yes. Okay, thank you.
Yeah.
But for me, I need to exercise more.
Yeah.
Yes, yeah, we can do it.
Yeah, you can. Yeah, I think. Yeah.
Refugio.
Well, I can't remember.
Refugio, or do you prefer? Refugio is asking if could erythropoietin also be a good candidate for neural regeneration, especially for patients with limited mobility.
Sorry, could you just repeat the name of the first part?
Erythropoietin. Erythropoietin
is something that enhances the production of red blood cells.
Yeah.
Yeah, it's like this.
Yes.
Yeah, is that, sorry. No, is that recently has been published some work that they are proving the erythropoietin as a option for the Alzheimer's disease.
Really?
So, but I don't know if you know something about some relationship with one of this improvement or some relation with exercise and erythropoietin production?
I would be hesitant to comment on that. The things we've looked at is how exercise promotes angiogenesis or vascularization in the hippocampus. I didn't show those data, but we see increases in, especially in younger mice, the increase in the diameter and length of blood vessels in the dentate gyrus of the hippocampus. So, and then there's quite a few studies showing, again, in humans, that there's the importance of increases in blood flow, cerebral blood flow with exercise, and how that links to cognitive function. So, but whether this compound that you mentioned has any of those kind of effects, I'm sorry, I'm not aware of that.
Okay, thank you. It was an excellent talk.
Thank you.
I will read some of the comments in YouTube. Victoria Brusolo says, thanks for amazing insight regarding exercise and brain function. Joel Omeli says, very interesting speech. Thank you so much. Aida Castillo says, thank you for interesting presentation. Marcos Nakmat say, excellent. Thank you for interesting talk. I don't know if anyone have another question. If not, Henriette, thank you very much.
Thank you again. Sorry.
I have a question, but if it's
okay, go ahead, Marta. Sorry.
Yeah, you know, my questions are all related to hormones because I work in that field. But you mentioned that estrogens have a role in neurogenesis, and so my question is: in menopause, there is a decreased cognition, and when the women are treated with estrogens, the cognition is facility are better. So are these related to neurogenesis? So I know that the rat, the mouse, is not a good model for menopause because they really don't have a menopause like the human, but do you have information about the neurogenesis and menopause and estrogens?
Again, my work or our work is mostly in animal models, but with so one of the things that was discovered is that during the estrus cycle, there is an increase in transient increase in cell proliferation when the estrogen levels are high. There's also a compound that I'm personally not that familiar with, allopregnolone. You may know better than me about this. So people have given that to animals and shown that there is an increase in so how do you say, in older female rats, I guess, there's an increase in neurogenesis if you give them allopregnolone. And that is now also in clinical trials for humans. So for postmenopausal women at risk for Alzheimer's disease. But I think the readout in the humans is cognitive function, not so much. I don't know what the brain parameters are that they are looking like. So yeah, so but basically the idea, or sort of, which is also sort of my philosophy on this, is that those compounds that work well in animals in terms of, you know, promoting neurogenesis, promoting learning and memory, could be good candidates for interventions in humans, regardless of whether there is a neurogenic effect. There could be a cognitive effect in humans with those kind of agents.
Thank you very much. Very much.
Sorry?
Thank you very much for your nice conference. And it's, I think we have to run instead of being all the time in the computer. Okay, thank you very much.
You're welcome.
Thank you very much for amazing talk. I hope we have the opportunity to invite you again in the future, and the better will be here in Mexico City.
Thank you. I'd love to visit, and I really appreciate being invited for your happy occasion for the anniversary. And I hope you have a good continuation of the festivities and of all the events.
Okay, many thanks, Professor van Praag, for this fantastic lecture. We are really grateful and delighted. You are always very welcome to our department.
Thank you.
Thanks a lot.
Thank you.
And we are gonna announce the next conferences on el martes 29 de marzo. Tenemos el tercer día de nuestras jornadas académicas con la presentación de la profesora Asma Nusrat, que nos hablará de plasticidad de las barreras epiteliales, y con el profesor Roberto Cosimo Melcangi, que nos hablará sobre el rol, el papel fisiopatológico de la neuroesteroidogénesis. Los esperamos el próximo martes. Muchas gracias y por ahora terminamos la transmisión en streaming.